the next of our microbes are the fungi. And here's an example of one of my favourite fungi, mushrooms. I quite enjoy eating them. And what I'm holding here is a thing called the fruiting body, or sporangium of a mushroom. So we do use them as food, particularly their sporangia. However, some of them are toxic, so you have to be careful of that. A lot of the toadstools and many mushrooms are actually poisonous to us because of the chemicals that they make as part of their life processes. Another really useful fungus is yeast. It's the fungus that we use to make bread rise and to brew beer and other alcoholic drinks, that sort of thing. It's used to make ginger beer because one of the waste products that it excretes is carbon dioxide, which makes those drinks fizzy. Also, one of the products that it makes is ethanol, which is alcohol. That ethanol isn't made during the bread making process, but it is for the alcohols, of course, that we use it for. So I'm going to use my mushroom here, because I'm hoping most of you have seen a mushroom, to show you the key physical features of a fungus and relate those to its life processes. So if I have a mushroom, it would grow away like this, and it's got a stalk, and the stalk is actually just a thing called a hypha. So it's not called a stem, it's called a hypha, which is the same word here. This is hyphae, the plural. There are two types of hyphae. There are the ones involved in nutrition, so they are inside the food source, whether it's bread, soil, whatever, and there are the ones above, and they are involved in reproduction. Underneath here, there's all these little lines, and in those lines, there are spores, which are a bit like seeds if it was a plant, but they're not like seeds. Seeds are the product of a thing called sexual reproduction. A male pollen and a female ovule, which is a plant egg, come together to make a seed. Whereas fungi use what's called asexual reproduction. They just make clones of their own DNA in the sporangia to make spores. So all of those spores are genetically identical unless there's been a mutation. So not surprisingly, these ones up here in the air, exposed, are the ones that this can burst or the wind can carry them away. This is how it does its reproduction. The spores float off until they find a new food source. They create some new hyphae. They start feeding. And then when they've done enough feeding, they create a reproduction hypha, and then that makes a sporangia that fills up with spores, and there's our life cycle. The most common one you probably see every day, hopefully it's not every day, but see regularly, would be that greeny bluey stuff growing on your bread if you've had it for too long. That's a fungus as well. And when you've seen the actual colour, you're seeing the sporangia, the bit that's exposed. It means that the hyphae are already all through that bread. So just rubbing off the top won't actually get rid of the fungus. It's actually all through the bread already with these nutritional feeding hyphae. In that, let's say it's bread mold, and I'm sorry I don't have some to show you, but if it's bread mold, for example, then what's happening inside the bread, so this will be in the bread, this is on the bread, in the bread, these hyphae, these feeding hyphae, are releasing enzymes that are breaking down the bread into simpler molecules and then absorbing it. We met this process when we learned about the bacteria. This is called extracellular digestion. So they have a couple of things in common with bacteria. They make spores which are identical clones, just like bacteria make identical clones themselves in a process called binary fission. They do extracellular digestion, just like the bacteria. But unlike the bacteria, they are what we call multicellular. They're made up of millions and millions of cells. They are sort of a community. As the hyphae spread, a new reproduction hypha can grow, and therefore a new sporangia can grow. And that's why you see fields of mushrooms, why you see so many or so much of that blue or green mold on your bread. It keeps spreading underneath the surface and then popping up these reproduction hyphae when it's time for it to reproduce and spread to a new food source. Now, fungi are actually really interesting things. They're good for food, but they're bad for things like athlete's foot, which is a fungal disease. They're bad for bread mold because it makes our bread waste, because the fungus is digesting our bread and sometimes making a toxic chemical, so a chemical that's not good for us in the process. They're also really, really good though, because they made the first antibiotics that we discovered. So the first thing's to kill bacteria. And the reason is they're often competing with bacteria for the same food source. So what a lot of fungi do is they create a chemical called an antibiotic, so something that will stop the growth of other things, to protect their patch so that they're the only organism eating in that patch, as it were. This was discovered a long time ago, that the chemicals that were made by some fungi would kill off bacteria, stop the bacteria growing there. We used this idea to develop our first antibiotics. We now make a lot of our antibiotics synthetically, so we do chemical reactions in the lab rather than getting fungi to do it for us. But fungi are fantastic because one of their life processes is to make a chemical called an antibiotic that we can purify and use to help treat bacterial diseases.